Good morning, students. We are going to start with the first chapter in our syllabus, which is anti-infective agents. So briefly, we will see what anti-infective agents and how they were discovered. So in 1867, Joseph Lister introduced antiseptic property principles by using phenol as a spray on an incision site and on wounds. In 1891 to 1900, Paul Ehrlich, microbiologist, discovered selective toxicity by using dyes and arsenicals. Prior to Paul Ehrlich's studies, local antimicrobial properties of phenol and iodine were well known, but systemic agents from Cinchona for malaria and Ipecac for amoebic dysentery were being also introduced. Ehrlich also discovered salvarsan as an effective antisyphilitic drug. Now, what anti-infective agents are and how they are classified. So first we will see what are the types of anti-infective agents. So anti-infective agents are of following types, 8 types. 1, local anti-infective. 2nd, preservative. 3rd, antifungal. 4th, synthetic antibacterials, wherein we are going to study drugs which are used to treat UTIs, urinary tract infection, antitubercular, antiprotozoals, anthelmintics, anti-scabies, and antipedicular. So in the unit 1 or topic 1, we are going to start with local anti-infective agents. And the definition of local anti-infective agents is, these are the chemical agents or the drugs which are used locally for infections and are called as local anti-infectives. When we say local anti-infectives, they contain 2 categories. One is antiseptic and second is disinfectant. Now, what antiseptics are? Antiseptics are the chemical compounds that kill or prevent the growth of microorganisms. So when I say kill, it will be bactericidal. When I say static, it will be bacteriostatic when applied to the living tissues, right? Now, any molecule, when we are going to use it on the cell or it will be used for pharmacological effect, it should have following good properties, ideal properties for a good activity. So what ideal properties an antiseptic should exhibit? So it should be rapid and sustained lethal action against microorganisms. It should be having low surface tension so that it can be spread into the wound. It should retain its activity in presence of body fluids. It should be non-irritating to tissues. It should be non-allergic. It should lack systemic toxicity when applied to skin, and it should not interfere with the healing property. These are the ideal properties. When it comes to disinfectant definition, these are the agents that prevent transmission of infection by the destruction of pathogenic microorganisms when applied to inanimate objects. Now, disinfectant also should have ideal properties. So it should have rapid lethal action against all potentially pathogenic microorganisms. It should bear good penetration power into organic matter. It should be compatible with organic compounds. It should not get inactivated by living tissues. It should be non-corrosive. It should be aesthetically pleasing, and it should be non-staining. Now, we will classify them. So the anti-infective agents are classified into following types. 1, alcohols and related compounds. The examples are ethyl alcohol, isopropyl alcohol, formaldehyde, ethylene oxide, and glutaraldehyde. Phenols and their derivatives. We have phenol, parachlorophenol, resorcinol, hexyl resorcinol, hexachlorophene, cresol, and chlorocresol. Oxidizing agents, hydrogen peroxide, hydrous benzoyl peroxide. Halogen containing compounds, we have tincture iodines and halozones. Cationic surfactants, benzalkonium chloride, cetyl pyridinium chloride. Dyes, we have gentian violet, methylene blue. Nitrofuran, we have nitrofurazone, furazolidine diones. Mercury compounds, nitromersal and thiomersal. Now, to understand what are anti-infectives, we will just quickly go through what infection is. So infection is the invasion of an organism's body tissue by disease-causing agents and their multiplication and the reaction of host tissues to the infectious agents and the toxins they produce. So this is what is infection. So anti-infectives is a common term used to describe any compound or agent that is capable of inhibiting the spread of an infectious organisms. So already we have classified them, and we are going to study each and every agent individually in our coming lectures. So classification in a nutshell, what we have already discussed in the chemical classification. And these are the structures. So here we will see the example of first phenols and their derivatives. So we have phenol. So every agent which is used to in any of the category will have its basic skeleton. So if we see phenol, so phenol will have a 6-membered phenyl ring with an hydroxyl group. When it comes to chlorophenol, we have an hydroxy group with an chloro group at the ring. We have metacresol, we have a methyl group, we have paracresol, and we have orthocresol. Basic skeleton of phenol remains the same. Substitution is done at varied positions, wherein the molecule is kept as it is and structural modifications are done to show the effectiveness of the molecules. Second comes the halogen containing compounds, wherein povidone iodine, a very well-known example of the ointment, and the second is halazone is there. Cationic surfactants, we have benzalkonium chloride and benzathonium chloride. Dyes, which are being used in the as a therapeutic agent, so we have gentian voided and methylene blue. Mercurials, we have nitromersol and thiomersal. Parabens, these are series of parahydroxybenzoates or esters of parahydroxybenzoic acid, and the examples are methyl paraben, propyl paraben, and butyl paraben. Alcohols and related compounds, so we have ethanol, isopropanol, formaldehyde, ethylene oxide, and glutaraldehyde. Phenols and their derivatives, we have phenol, parachlorophenol, resorcinol, hexyl resorcinol, or hexachlorophene. We have cresols and chlorocresols. Oxidizing agents, H2O2 and hydroxybenzyl peroxidase, halogen containing iodines and halozones. Cationic surfactants, the structures we are dealing with. Dyes, we have already mentioned, the nitrofuran, the mercury compounds. So we will see the mechanism of action of alcohols and related compounds. So here we have non-polar and polar groups. Primary alcohol shows more penetration, more active, more cost as the length of the carbon chain increases. So the best example to quote here is N-propyl alcohol. So primary alcohol shows more penetration and they are more active, more cost effective. And when we are going to change the carbon chain, we are going to increase the efficacy of the molecule. Secondary alcohol shows less penetration capacity. They are less active. Example is isopropyl alcohol. When R increases, when I say R, it means the non-polar group. It increases the penetration, it increases the action, as more van der Waals forces are present, and therefore the pharmacological desired action is going to get reduced. So understand, when non-polar groups are being attached, are being increased, it is going to increase the penetration power, but thereby it is also going to decrease the activity what we are talking about. Formaldehyde mode of action is alkylation, which alkylates the groups present in the proteins and the nucleic acids. As the primary alcohol chain length increases, van der Waals interaction increases, and ability to penetrate the cell membrane also increases. So therefore, as water solubility is going to decrease, the antimicrobial potency decreases with the decreasing molecular weight. So branching of the alcohol chain decreases the antimicrobial potency. The germicidal action of formaldehyde is slow but powerful, and the mechanism of action is direct non-specific alkylation of nucleophilic functional groups like amino, sulfadryl, hydroxyl present in proteins and nucleic acids to form carbonyl derivatives. Next molecule, next examples of phenol. Phenol in low concentrations denaturate the proteins, the bacterial proteins. Phenols in high concentration produce lysis of bacterial cell membrane. Mechanism of action of oxidizing agents. So oxidizing agents are going to liberate oxygen, free radicals. Anaerobic microorganisms are affected by these liberations and they will die. The germicidal action of oxidizing agent depends on their ability to liberate oxygen in the tissues. All these agents react in the tissue to generate oxygen-free radicals, and these agents are effective against anaerobic microorganisms. They are used in cleaning contaminated foods. Next, mechanism of action of halogen-containing compounds. So we'll take example of tincture iodine. Iodine will inactivate proteins by iodination of aromatic residues like phenyl, allyl groups, and tyrosyl groups. Iodine can oxidize SH group by oxidation of sulfur-containing groups, and chlorine-containing compounds will release chlorine and chlorinate amide nitrogen atoms so that amido N2 is halogenated and also oxidation of sulfur-containing group occurs. Cationic surfactants, will act by dissolving and disposing into the cell membrane and then destabilizing and causing lysis of the cell membrane. They will interfere with the enzymes present in the cell membrane. Thereby, it is going to show its action. Dyes will exhibit a mechanism of action on cell membrane, and the difference in susceptibility of the organism is related to cellular characteristic. These cationic dyes are active against gram-positive bacteria and many fungi. Gram-negative bacteria are generally resistant. Nitrofuran derivatives, the complete mechanism is not yet understood, but they are mutagenic and carcinogenic under certain conditions, and the cellular effects are due to DNA damage caused by metabolic reaction products of the nitrofurans. This particular thing is going to help us to understand how the mechanism we have studied, and the mechanism of action had explained us about the different derivatives, how they are going to be used. So this was all about the mechanism of action and therapeutic uses, a very small chapter named as local anti-infective in our syllabus. Now we will be proceeding towards the next chapter as preservatives. Thank you.